Hello everyone. Today I'm going to talk about unusual benthic fauna associated with a whale in Monterey Canyon, California. What is the whale? When whales died and their carcasses sink to a bottom of the sea known as whale, it provides a abundance of nutrients for deep water creatures. Whale falls along the California margin suggest that community succession through three stages. First, the mobile scavenger stage, comprised of about 40 species, mostly fish and crustaceans, remove more than 90% of the soft tissue, usually within 4 months after arrival on the seafloor depending on the size of the carcass. Second, the enrichment opportunistic stage, comprised of about 20 species, mostly polychaetes and crustaceans, may last as long as 2 years. Finally, the zoophilic stage is dominated by microbial mates and more than 100 invertebrate species, may last for years and even decades depending on the size of the whale. The skeleton was at 2,891 meters depth in the axis of Monterey Canyon, the red star in the picture. It's one of the deepest whale falls discovered to date. The surrounding floor of the canyon was composed of fine-grained karstic sediments. Trash and seagrass were observed near the fall, suggesting that this is an active sediment area. Based on the number of whale falls discovered during the past several decades, these habitat islands may occur relatively frequently in time and space. So we are wondering if organic enrichment due to whale falls could play an important role in regulating animal abundance and diversity in deep regions. Canyon exploration, sample collection, and video surveillance were accomplished using ROV tubular. Sediment push cores, bone samples, and animal specimens were collected with the manipulated arm of tubular. Video recordings, the data of abundance estimates, and bacterial mat coverage areas were taken from the tubular using two different digital cameras. Animal identifications were accomplished through morphological and/or molecular characterizations. The whale was identified as a gray whale, based on diagnostic building structure and confirmed by 99% DNA sequencing similarity of known gray whale. And this is Berlin. The length is about 9 to 10 meters. Based on weight to length connections, the biomass is about 20,000 kilograms. Because gray whale reach sexual maturity at 11 to 12 meters in length, so this specimen was assumed to be a juvenile. This animal may be dived during the round-trip gray whale migration from the food-rich Arctic, Bering, and Chukchi seas to the breeding grounds of Baja California. At Taiwan, the remains of the whale include flesh, intestine, and belly. The skeleton was intact, suggesting relatively recent arrival on the seafloor. Bacterial mat was developing near the skull, maybe promoted by sulfide produced from microbial decay of lipids. The dominant animals associated with the whale were two unusual polychaetes, Annelid A. occupies a basal position among members of family, with only 85 to 88% aiding that DNA sequence similarity to other symbolic genera. The 12 to 15% difference between Annelid A and other members of the group provides good support for the novelty of this species. Annelid B. The analysis of ribosomal 18-net sequences revealed that Annelid B showed 86% similarity to two selected genera of the family Spionidae. It's likely a new species or even a new genus within the family Spionidae. Other species observed from the whalefowl. Polychaetes, such as large green tube-wearing amphitrachs, dominate the near sediment community. It's worth noting that many of these polychaetes are thought to be new taxa. Second, decapod crustaceans appear to dominate the scavenging component of the whalefowl. Time 2 is 1 month after Time 1. The whale skull was still easily recognized, but wrists and other skeletal parts had collapsed to the substrate. Flesh and intestines were not as prominent as at Time 1. Associated fauna at Time 2 was very similar to Time 1, including the dominance of annelids A and B. The area covered with bacterial mats had increased. The area changed from 1 square meter to 6 square meters. Time 3 is 8 months after Time 1. Very little soft tissue remained, and many bones were degraded. Coverage by bacterial mats had greatly expanded. The area changed from 6 square meters to 15 square meters, and was heaviest in sediments near to the carcass. Underlying and near sediments were black, and core samples smelled strongly of sulfide. Annelid A still appeared to be the dominant animal. Annelid B, which was abundant on previous species, was nearly absent. New animal species not observed at time points 1 and 2 include large polynoid polychaetes, 3 species of Sipunculus, and large nematine. Glassite crab and large amphitrachid polychaetes dominated the surrounding sediment, which were in much lower abundance than at times 1 and 2. Individual whalefowl communities may differ greatly depending on the location, depth, and the group of available colonizers. We assume that the delayed degradation of Monterey whalefowl community results from its depth. Increased pressure and low temperatures may promote organic preservation. Annelids A and B at whalefowl appear to be specialists. Exclusive use of such temporary habitats would require rapid growth, early maturity, high reproductive rates, and greatly dispersal capabilities. Monterey Canyon is an active sediment transport conduit that promotes the rapid movement of recent sediments into the deep sea. The deposition of large animal remains in this transport conduit has the potential effect of diversity and structure of benthic communities. There are three conclusions of this paper. First, we found the soft tissue of the carcass will gradually disappear. At time 1, the soft tissue includes flesh, intestine, and spleen. At time 2, the remaining soft tissues were not obvious than time 1. At time 3, just very little soft tissue remained. Second, the area of bacterial mass will increase from 1 square meter to 6 to 15. Finally, two new species were discovered, annelids A and B. Annelid A was found only on bones. Annelid B was in tissues and surrounding sediments. Thanks for listening.
Thank you for your presentation. I think it's quite good, but uh, no, but you add a lot of explanations than before. But I think you should like look at the audience more. Like you should be doing a presentation instead of reading the PowerPoint. Yeah, but I think it's good. Yeah.
Thank you, and it's good to see that you put a lot of images, especially you put vivid pictures of welfare in the like very first of the page. So it's good to lead the audience into the topic, and I think this little I'm impressive with that you put the little word box in the corner of the slide so that it remind us of the information that we talked before. And I think there is a little suggestion is it is important to keep eye contact with the audience so that the audience can follow you easily. Thank you.